Good morning, everyone. Welcome to our session. So first, we would like to welcome Dr. Michelle Campbell. Is she on Zoom yet?
Megan, can you hear me there in the conference room?
Yes, I can hear you, Dr. Campbell.
Great.
I think we're working on getting everything queued up with the screen. Okay, Dr. Campbell, we can see your slides now.
Great.
All right, you can take it away.
All right, well, thank you. Well, good morning, everyone. My name is Michelle Campbell, and I am the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience for the Center of Drug Evaluation and Research. And so I want to thank you very much for your invitation this morning to talk to you about the drug development process from the FDA perspective. I want to apologize for not being there in person, but I am thankful for this virtual option so I can spend a few minutes to talk to you about what happens with the drug review at FDA. So typically, I spend a lot of my time presenting on patient-focused drug development, clinical outcome assessments, rare disease, clinical trials, and the challenges with those. And so today, it's actually a great opportunity for me to speak about something that is actually at the foundation of everything I feel like I talk about is really what do we do every day? What do we do at the FDA as reviewers? What are our rules and regulations? And so I think this is a really great way for you to learn about that, and hopefully I can answer your questions. But I think it's also really important for us to talk about how the FDA even started. How did we get these rules and laws to pass for us to do the jobs that we do? So if I can have the next slide, please. The modern era of the FDA really starts back in 1906 when the Pure Food and Drugs Act was passed. This was an act that added to a regulatory responsibility to the agency's scientific mission. And it prohibited interstate transport of unlawful food and drugs under a penalty of seizure. But it's important as it was, it had some shortcomings because it didn't have the authority to regulate medical devices or cosmetics. And it really didn't give the FDA any authority to inspect factories or to prosecute any false therapeutic claims. And it didn't give us the control over what drugs could be marketed. Next came along in 1937 was really that defining point in FDA history. And I'm sure many of you guys have heard the story in history about in 1937, there was a company that was trying to develop a anti-infective medicine, a cough syrup, and it introduced the elixir of sulfonilamide that was a flavorful liquid, and it was great, except it had in it an untested solvent of diethylene glycol, also known as the main chemical in antifreeze. And unfortunately, the FDA was not aware of this problem and was unable to remove the product before it had caused the death of hundreds of children. And so when this happened, it really invigorated a bill in Congress to replace that 1906 Pure Food and Drug Act, which had really been, we were hoping for this new bill to pass, and it had taken some time. But in 1938, we finally had the passage of the signed Food, Drug, and Cosmetic Act into law. And so really that's the first time we even got to think about looking at safety of drugs. And I think it's really important is that the Food, Drug, and Cosmetic Act from 1938 is still a lot of our foundational regulations are added onto that, but that is the basis of the work that we do every day. And so what it did is it gave us oversight of cosmetics and therapeutic devices. It required that new drugs be shown to be safe before marketing, and therefore we really started this new system of drug regulation. And it authorized factory inspections and allowed court injunctions in addition to the previous penalties of seizures and prosecutions of things that were not had not been regulated. But it's really important that this is the first time we really were like, we need to look at safety of products that people are calling a medicine. We move forward to 1962, and it's actually where we come a lot closer to what we do every day now through the Kiefhofer-Harris drug amendments to that Food and Drug and Cosmetic Act. And this was because it came on the heels of us preventing another incident of potential death from a drug or product. I think a lot of us know about the story of thalidomide, which was actually a sleeping pill that was caused to found birth defects in Western Europe. And it was particularly being given to mothers who were pregnant at the time. And so there was a new medical officer at the FDA. Her name was Dr. Frances Kelsey, and she was reviewing that drug application at the time and noticed these issues and really fought for us not to approve this marketing application. So at the time, in 1962, was finally when the act was passed, and it was gave us to ensure now efficacy as well as greater drug safety. So for the first time, drug manufacturers were required to prove to us that products were not only safe but effective before they were able to market them. And this was really what we do every day, right? Our job is to make sure that drug products and treatment options are safe and effective to be able to put onto the market. I'm also quickly going to touch on about biologics because of the Greek. I just learned some new names, and the GRIN disorders, because of the genetic nature of them. I'm sure folks are also thinking about, could there be a potential for a gene therapy at some point? So I'm just going to quickly touch on some brief history of the regulation of biologics from our Center of Biologics that we have. So modern oversight of the biologic products began in 1902 under the Biologics Control Act, and it passed to ensure the purity and safety of serums, vaccines, and similar products used to prevent or treat diseases in humans. And this was carried out by the Hygienic Laboratory of the Public Health and Marine Hospital Service. With the creation of the National Institutes of Health from that laboratory, the regulatory authority was actually with NIH until 1972 when it joined the FDA, and that is now under our Center for Biologics. Next slide, please. So there are 3 medical product centers at the FDA. And so I want to quickly just introduce them to you and highlight what they each do. So our first center you see is our Center for Devices and Radiologic Health. And they are our center of devices regulates a lot of things. They regulate from, something as small as maybe we heard a lot about masks during the pandemic, to laboratory tests, to actual pacemakers. So they cover a wide range of things that they regulate, and they have specific regulations related to them on their approval processes, and clearance process. They clear devices as opposed to approve them, and so their regulations are different than the regulations for both drug and biologics. And drug and biologics follow the same regulations. The next center is our Center for Biologics, or CBER. CBER is our center that does gene therapies. They do blood products, they do vaccines. And so there's definitely great interest in a lot of our rare diseases. For our new Office of Therapeutic Products in CBER, particularly that is where the gene therapies are housed. And so I do know that for our rare disease families, they are very much interested in the efforts that CBER does. And both CDER and CBER collaborate frequently. Actually, all 3 of our medical product centers are in constant communication with each other and collaborate all the time on various topics, issues, particularly when it comes to patient-focused drug development. We brought in that to actually patient-focused medical product development as well. So we do talk to one another. And finally, I want to talk about the center that we're here to talk about today, which is our Center for Drug Evaluation and Research, or CDER, which is where I'm based in. And so in CDER, we have many super offices, as we call it, that break down to our review divisions. But the one that I think we're really here to talk about is what happens in drug reviews, which is mainly based out of our Office of New Drugs, which is where we review investigational new drug, and then finally programs, and then finally new drug applications or biological license applications, which is really that marketing application that we review in order to make an approval decision of whether a medical drug can be marketed to the US population. Next slide, please. So what happens in drug development? I want to note that the FDA is just one stakeholder when it comes to drug development. There are many stakeholders that are involved, and I have a good feeling that a lot of them are in the room with you today. It includes patients, it includes caregivers, it includes academics, it includes industry. And so together we are all stakeholders. I also want to let everyone know that FDA does not actually conduct the clinical trials, right? That is what the sponsors and industry do. That's what academics or academic clinical centers do, is conduct the trials. What we do is we review the evidence that comes in, and we provide advice and make decisions based on that evidence. But let's start a little bit back before we even get to having data to be reviewed. How does the actual drug development process start? And it really starts early with drug discovery and development. And so what happens in there is this is when a lot of the bench science and a lot of what are probably our academic partners that are there with you today spend time looking at understanding the disease, the natural history of certain diseases, looking at mechanistic aspects of not only the disease, but is there a molecule that perhaps could impact the aspects of the disease because of the mechanism of action. They may look at molecular compounds to find possible benefits, and really just looking at what is something that could be carried forward and tested to be moving forward that may have promise. And so this can take a long time, but it's a lot of this bench science that we talk about when we think about translational science that really is looking at what is something that could potentially impact the disease course of a specific disease. The next step is our preclinical research, and this is done either in vitro or in animal studies. And this comes before any type of human studies and consists of various tests to evaluate safety. It provides information to support initial human doses of a drug. If a drug is planned to be used in special populations, such as pregnant or breastfeeding people or children, there's additional studies that will be required. And so with that information in hand is really trying to see, is this compound safe enough that we think that we want to test in humans? And so when we get to this part where we're looking at first in human studies is when we typically get involved in the investigational new drug process, when a sponsor will submit their IND application to us and really begin that drug development process. So when we talk about clinical drug development, it's defined as studying a drug in humans, and it's conducted in a sequence that builds on the knowledge accumulated from our non-clinical studies and that previous any clinical work or anything smaller studies that would have been informative. The structure of drug development programs is shaped by many considerations and comprise studies with different objectives, different study designs, and different dependencies, so that every phase that we're going to talk about may be distinct or will be seen in a program. But this is just a general overview of drug development. So the purpose of the first phase of clinical research is really to look at the safety and tolerability of a drug. And this is often known as a phase 1 study or perhaps a first in human study. And these are really important for us to try to see what that is, that tolerability. And we're looking at perhaps a dose range. We're looking to make sure that it's safe in humans. And so this is where in drug development, we often find this in healthy adults or healthy volunteers, I should say, to test this in. If these studies they're typically done in single doses or perhaps small multi-dose range studies to see what kind of safety and tolerability. We also start to look at something called pharmacokinetics of the drug, or what does it do on the body. And this is how it helps us understand how the drug is absorbed, distributed, and metabolized and excreted from the body. And during this phase, we also get information on something called pharmacodynamics of the drug. And that, again, looks at what is the impact on the body as well. And really, this can help us estimate I guess in a way, how we internally in our bodies, we can't really see per se, the activity that will guide us in dosing potential future dose regimens to test. If these are successful, this will move then to the next phase of drug development, which is phase 2. And so approximately 70% of drugs are moved from a phase 1 to a phase 2 part. So after these initial clinical studies, that provides sufficient information on safety and clinical pharmacology and a potential dose or doses to explore, we look at exploratory and confirmatory studies, typically referred to as phase 2 and phase 3 trials. And they are continuing to evaluate safety and the efficacy of the drug. So in our phase 2, or sometimes our exploratory studies, they're typically designed to continue to look at safety and efficacy. And this is when we're going to start to look into that actual patient population that we want to study. At this point, we believe that the drug is safe to be given to humans, but we are constantly looking for safety and following it over the course of the drug development program. And we want to see what the drug is actually doing. So in these studies, we're really refining the dose of the regimen. We have a sense of what's tolerated, but we need to know what is actually going to be effective in treating. So is there a specific dose level that needs to be used? And it allows us to do dose ranging studies to really explore that. We also use this to clearly identify the target population of interest and who should be receiving this treatment option. Additionally, we're capturing more safety. We're capturing more of that pharmacodynamic data of really how is it being metabolized in the body and excreted. And so these are often really informative. This is what's going to inform a successful phase 3 or confirmatory trial. And so about 33% of these drugs will then move into a phase 3 trial setting. So our confirmatory studies, also referred to as phase 3 trials, are our pivotal studies that are usually intended to provide the adequate basis for marketing approval. They're also expected to provide information that supports the development of both adequate instructions for use of the drug and the official product information that would be submitted in a marketing application. These studies look at that patient population that would support the marketing of the drug. And so these are what is described in our product label, and these are rigorously designed studies that are well conducted, that have pre-specified statistical analysis plan that will be reviewed by us and assess whether they have the evidence to support an approval for a marketing application of the product. In these studies, we are continuing to evaluate safety. Safety is always at the forefront of our mind, but really efficacy is also obviously extremely being looking at. And this is where we talk about what is going to be your endpoint. How are we going to assess that endpoint? Does it make sense to be assessing that endpoint, or are we missing something? It's expected that these studies will be, the length of these trials will be dependent on how long the drug will act in the body. And so if we were talking about something that was an acute condition, perhaps we may have a shorter trial design. But if we're talking about something that may be a lifelong therapy or treatment, we may be looking at a longer study design and trial length. And that also, these are all dependent on a lot of other factors. And these are the conversations that a sponsor will have with their respective therapeutic division, what that would look like. And then these studies, because they're longer than our phase 2 and phase 1 studies, this is where we get a lot of our long-term safety information, and we're building our safety databases. Finally, if we are successful and are able to approve a marketing application, so after a phase 3 study is finished, a sponsor will make a decision that they want to submit a marketing application for review. And then during that process, the agency will review that marketing application, and we have time clocks of timelines of how long it will take for us to review. If we do ultimately approve that marketing application based on the evidence that's been submitted to us, there could be what we call post-market or phase 4 trials. And so these may answer certain questions based on information we want to collect some additional information to help us. It could be, again, on safety. It could be for some other things. It may be to confirm some requirements in law of certain studies, smaller studies that we would need that were not completed in that initial development program. Again, this is all determined based on that marketing application review of what would be needed, and these would occur if there had been a favorable marketing application decision. Can we please have a next slide? So often a question I get a lot is, well, who makes up the review team? And I must say that that any decision, advice given is not by one single person. It is made by a group of folks. There's multidisciplinary teams that are part of our review team. And this is just an example of some of those who are part of it. And they are part of this process through the entire drug development program. So first, when an investigational new drug application comes in, to all the way to that marketing application. And so we have a project manager who is actually coordinates all our activities. They are really the person that makes sure we meet our deadlines. They are the person that really that person that interacts the most with not only a sponsor, but with all the other team members within the FDA. We have medical officers who are typically physicians who review all the clinical study information and data that is coming in for the evidence. We have statisticians that look at the study designs, they interpret the trial data, and they work very closely with that medical officer to evaluate the data that has come in. We have pharmacologists. I talked about clinical pharmacology and pharmacokinetics and pharmacodynamics. And this is the group of people that review those type of studies. We also have folks that are non-clinical folks that review all the animal model data and the animal studies that come in, and they have some different training of being either maybe a chemist or a pharmacologist as well. We have folks that are chemists and microbiologists and manufacturing people. So those folks look actually, our chemists and microbiologists will look at the actual compounds that make up a drug. And they're looking to see how the drug is made. Is it stable? Is there any impurities we need to be worried about? They look to make sure there's no cross-contamination, or that like if it's a microbiologist, that there's no any type of antimicrobials that may maybe develop in terms of that development process of making the drug. We have manufacturing experts. So one thing that we do when we approve a drug is we actually approve the manufacturing of the drug. And so that's a really big deal as well. So we're not just approving that the drug works, we're actually saying that the manufacturing of it is up to standard and up to good clinical practice, or good manufacturing practices to be able to manufacture this drug. And we have safety folks who look at safety data. We have inspectors that go out and investigate sites. It's a big team. We have a lot of subspecialties. We have a lot of internal consultants, such as my former life when I was reviewing clinical outcome assessments. We are a whole host of multidisciplinary folks that come together to review and provide the best advice to a drug development program. Next slide, please. So now I've filled you in on some of the history and who works on these teams. I want to talk a little bit about some of our actual regulations and how we have to think about the evidence when we're making our decisions to market an application. And so I'm going to start talking about, the level of evidence in our regulations. So the FDA requirements for drug approval are documented in the Code of Federal Regulations. So everything we do is mandated by this regulation and law. In general terms, the approval of an application requires that the application meets the requirements of safety, efficacy, chemistry, and manufacturing controls. So different disciplines within the agency will review these broad areas. And so I'm not going to really be speaking about the manufacturing aspect today, but I will talk about more of the things what I would focus more on is really that aspect of efficacy and safety. So the efficacy standard that we abide by states that the program must provide substantial evidence consisting of adequate and well-controlled investigations, and that a drug product will have an effect on of its purports or are represented to have under the conditions of use, prescribed, recommended, or suggested in the proposed labeling. So this is that's part of the big law of what it says. So efficacy is our standard. And so what we really want to do is, do we have evidence that the drug works, or as in, is there a clinical benefit where we have an improvement in how a patient feels, functions, and survives? And so that is what we're going to be looking at with our efficacy data. And so depending on the disease area and the number of patients available for disease, where we talk about that, what substantial evidence, which is not going to be a point that we're going to be discussing much on today. It will vary. And again, this is why we encourage sponsors to come talk to us early to have these discussions about what can really be done and captured in their program, particularly in our rare disease programs, where we know that we don't have many patients and we want to make sure that we're getting the most information from our rare disease populations in our trials and not really having to really, we want to make sure that we're using our rare disease patients to the best we can and not really putting them through trials that will not show efficacy. So we're really considerate in all of our trials, but when it comes to our rare diseases, we understand that there's always going to be development challenges. And so we want to make sure, how can we optimize our approaches to rare disease drug development? And so what we talk about in the broadest sense of when we're trying to apply efficacy and looking at the evidence, it means that we are trying to typically interpret two adequate and well-controlled trials. But in cases, again, this is where is there sufficient information from one robust study and additional supporting evidence, confirmatory evidence. And again, this is a discussion that industry and sponsors will have with their respective therapeutic division about what that looks like. So when it comes to safety, the Code of Federal Regulations really discusses the factors that would cause the FDA to refuse an application based on safety. The safety standard explains that an application would not be approved if it did not include all the adequate tests by methods reasonably applicable to show whether the drug is safe for use, and if the results of the test show the drug is unsafe for use, or if there's insufficient information to determine the product's safety. So the safety standard is a little bit more general, and which is why we closely review all of the safety data that is provided to us to make our safety determinations. And we use this information as part of our regulatory framework when we're making those decisions, whether or not a drug has met both the efficacy and safety standards set up by the Code of Federal Regulation. We take all of these things together and really try to make a benefit risk assessment on the things that patients will see when we are reviewing our new products. And it's really the push to summarize that benefit risk is, and what is that balance of our reviews. So for the next slide, I really want to talk a little bit about benefit risk. So this is our benefit risk framework. And so what we're trying to do when we are reviewing a marketing application is really try to find a balance between benefit and risk. So what does the drug do? And is there risk? And that's the safety aspect, right? And so we really are looking at if there is a safety, event, perhaps, is this something that we expect? Was it because of the drug? These are things that we're going to investigate to make sure that we feel that it's safe enough to be able to put on the market and it will not do harm to patients. And so this is what we think about. If you look online at any of our approved new molecular entities or the drug labels, you will see these in our reviews, this table, and it outlines a lot of things. What is the current, what do we know about the condition? What are their current treatment options? What is the benefit? Can we mitigate risk? How can we do that? And so it really is showing that balancing and the thinking of our approach of how we're really thinking about this framework. And so it really summarizes the benefits, the risks, who the patients are. And I think it's really important to know is that we often hear from patient groups and patient families through listening sessions and patient-focused drug development meetings. And this is where that input that we hear from you, and I talk about this a lot, that sometimes often asked by patient groups, how do you take back what you've learned from us? And I often say, well, sometimes that is not immediately seen because if there may not be a product available for us to review yet. But this is where this input comes into play. This is where we take what we hear from you. This is one way where we take what we hear from you and we put it and think about it into our benefit risk framework. So we are listening, hearing you. We also take that information from those sessions into the advice we provide and the considerations and things of that. But this is definitely one example of how we do use that information from patient-focused drug development, from listening sessions, and from meetings like this that we hear and learn from patients and their caregivers. Next slide, please. So quickly, I just wanted to talk about a couple designations that you may hear when it comes to drug development. So first is orphan drug status. And so we obviously know that developing any drug is expensive, and we know that doing drug development in rare diseases can be a challenge. And so orphan drug status was established in the Orphan Drug Act, which I believe we are celebrating, I think I'm going to say the wrong year, 40th, right? Am I doing my math right in my head? Anniversary of it. And so the orphan drug status or designation is a program that provides a designation of that in their treatment. And so it does put a number that it's available to drugs where it's intended for the safe and effective treatment by diagnosis or prevention of rare diseases or disorders with fewer than 200,000 people in the US, or to treat a condition that affects less than 200,000 people, for which there's no reasonable expectation that the sales from the drug will be sufficient to offset the cost of developing it. And again, this is for marketing within the US. When this status is conveyed to a drug, it's not just about the drug, but also the disease it's treating. And so this, again, like I said, came from the Orphan Drug Act. And if my memory serves me right, this was passed in 1983. This was a really big deal. And so this provides incentives to sponsors to consider developing drugs in the rare disease populations. And so during a drug development program, a sponsor can apply for that status, and that is handled through a different office or Office of Orphan Products with that. Next slide. And I also want to quickly highlight expanded access. Expanded access is a program that people hear about. It is a program that we've had for many, many years at the FDA. And the idea is that expanded access program is for people who are not in the current clinical trial, but can have access to an investigational drug. And so there's procedures in which how that is. A person has to reach out to a drug company that has the drug to see if they would consider it. The drug company submits information to the FDA. The FDA makes a decision about that. But there is options, and this is one option. Again, it does fall back a little bit onto if the sponsor can do it or not. But we do very much are supportive of these programs. And I'm going to end with one last slide. I realize I've talked really, really long. And so if I can have the next slide, please. I want to tie this back into patient input, as this is a conference with, I know, a lot of patients and caregivers there. Patient input and patient-focused drug development is the way that we review and think about drug development. And patient input is critical at various stages of the drug development process. As I said, FDA is just one stakeholder in drug development, and drug development is really a spectrum because FDA only comes into play once we have a first-in-human trial. And so I encourage patients to understand that their voice can be incorporated throughout the drug development spectrum. It can be discussed early when we're thinking about the impact of the disease and what matters most to patients and how to measure them. In our clinical trials, patients can help with suggestions of clinical trial design and things of that nature. And I think about a trial I was a part of as a research coordinator, where we had to remove someone from the study. They asked to end the study early because the timing of when the drug was being administered was during lunchtime. And that was an important aspect to their child's life. And they did not want them to take away from their friends at lunch. Had that person, and I was, like I said, I was a research coordinator, so this was an academic study, and had the main PI thought about that and we talked to patients, we could have thought of perhaps of a different way to make that approach. And so I think patients are important, can play an important role during the entire process. As I mentioned, how patient data can help us when we're reviewing a marketing application. And then finally, how do we best communicate this information? So I want to thank you again for allowing me to come today and speak to you. I greatly appreciate it. I know this was a quick overview of the drug development process from the FDA perspective, but I'm sure that we will have continued conversations in the future. And so again, thank you.
Thank you so much, Dr. Campbell, for a great talk and revealing what the regulatory process looks like for drug development. For everyone at home, remember that you can check the dialogue section on the left-hand side. If you're on a web browser to input your questions, those of you on the mobile app, you can go to the communications section. We've already received a couple of questions. So our first question for Dr. Campbell is, are there specific patient numbers required for safety and efficacy approval? With our smaller numbers, we want to make sure that we balance this without waiting forever, as our kids are at risk without treatments. Dr. Campbell, are you still there?
Yeah, sorry, I muted myself and then I couldn't unmute. Sorry about that. Technology is great on the Saturday morning, everyone. So that is a really great question. And so I want to say this, I don't want to put them out here and say there's a specific number. This is a great question that we definitely take to matter about knowing how many patients do we really have. So I would say in two parts. One, it's really knowing how many patients do we really have in a disease and a disorder, and what is the likelihood of us recruiting how many patients into a trial. The second piece is, this is where your industry colleagues, and I know that you have someone speaking next, we'll have these conversations actually with the agency about what kind of numbers do they need for safety and what would that look like. And so I think there's a lot of way where we can apply this as some flexibility on what that is, but those are going to be very much drug development program specific, which is why we don't really want to give a specific number at this point, because it's going to really depend on the condition, what the drug's supposed to do, what does the safety profile look like? So it's very, very dependent, but this is a question that we handle and we deal with all the time. So we're not unfamiliar with thinking about this. And again, this is a really great conversation that we know that typically industry will come and talk to us about. And so we try to see what are our options and what it could look like. So we do take into account how many patients, are really available for us to be able to capture in a trial. And we do understand that.
Thank you, Dr. Campbell. Our next question is, what is the average caseload for FDA clinical trial reviewers at CDER? How many trials are you considering or monitoring at any given time?
Wow, that's a really great question. I don't even think I have an answer to that, because that's all we do, right? So, I think it's really, I really appreciate that question, because that is a really fascinating question that making me think about how many INDs we have and how many meetings I have a week. So basically anytime, someone is investigating a drug, we should be involved, right? This is when an IND. So I can't say what our caseload is per se, because it really is dependent about when does someone come in and talk to us so we can provide advice. And so someone may come in, and so we're active at that moment, and we're getting continued updates. There's requirements to report safety and reports throughout the development program, but it could take time. We may not hear back from them for 6 months to a year or so, because they're doing their trial. And then when it's appropriate for them to come back to get that next device, they're back. So I really don't think we can give you a hard and true number, but we're constantly looking at that. In terms of, I can tell you this, in terms of approvals in 2022, for new molecular entities, so those are drugs that are maybe first in class, the first new drug for a condition, for example, or something. So these new molecular entities, we approved, I think it was 54 last year, and over half of them was in a rare disease. And we've had probably 4 rare disease approvals already this calendar year. We also have something called supplements. And so that's when, which is not, that number is a little harder to track, but supplements are when maybe there's a drug that's already been approved for another disease, and it's now has a new, what we call an indication. So we found that it's safe and effective for a new population. And so that's another way for drug approval for an application. But I really can't say, because it's always a very active space. I would say that we are always very busy. And we're always making sure that the advice we give is to make sure that we have safe and effective products, and that we're making sure that we're giving the best advice to make sure to assess those.
Thank you, Dr. Campbell. We'll take 2 questions from the floor, and then we'll move on to our next speaker.
Hi, Michelle. This is Tim Benke, University of Colorado. Good to hear your voice again. So my question is if you could address to this group how important you think it is for them to set up a patient-focused drug development meeting with you and what that time frame might be.
Hi, Tim. Nice to hear your voice as well. And great question. I'm sorry I'm not there to see you in person as well. And I think it's a really great question, Tim, because I know Tim has been a part of a couple of our patient-focused drug development meetings. So it is extremely important. We very much enjoy learning from patients and hearing from your lived experience and the caregiver lived experience. And so there are multiple ways to come and engage the agency. First, can be a simple listening session. That can be coordinated through our Office of Patient Affairs. It's a much shorter session. It's about an hour and a half, and it's really there for us to get that maybe first exposure of a disease and disorder and that lived experience. But the next step that a lot of patients groups consider is really our externally led patient-focused drug development program. And so we are in PDUFA 7, so in PDUFA 5, back in 2010, we were charged with hosting patient-focused drug development meetings. This is where patient-focused drug development really began, the era of it began. And we successfully hosted meetings, but we found them extremely valuable. And while we were no longer mandated to continue having these meetings, we still have had these meetings. So the FDA will host meetings now that may be of an important emerging public health issue, like the upcoming one on long COVID in April. But what we started with is this external one, which is where where patient groups will actually host externally led patient-focused drug development meetings. And what they do, they submit a letter of intent to our patient-focused drug development group out of the Office of Center Director. And they submit a letter of intent about what they want to do and what they want to cover. And then I can say that then that letter of intent they will reach out to us and the review staff in this respective therapeutic division about, do we agree that this is something in an ON? We are like, yes, we like to hear from patients. And then a meeting will be set up. And so it could depend on the timeline. It could be 6 months, 9 months. It depends on when that letter of intent goes in to planning it. But typically these are all-day meetings. And this is where, again, more FDA staff attends. We listen, we learn of that, and you can cover a lot more broadness of the disease aspects than a listening session. So if you're wanting to consider one, you can Google, because that's how I find things on our website myself. If you Google PFDD CEDAR or PFDD FDA, you'll actually come to a landing page for our patient-focused drug development group, and it will have the information. There'll be 4 blocks, and you'll see one that says externally led patient-focused drug development meetings, and that will contain the information on how to apply and submit a letter of intent.
Thank you. All right, one more question from the floor.
Yeah, I had a very specific question. So I know a lot of parents are interested in L-serin, which is a food supplement. It looks really safe, but with very small effect. And the population that we have has really a high variety of symptoms. And it's very hard to set up a double-blind study for that. I was wondering if you have any advice to our community on how to approach that we set up a study that it will be funded in the end and approved by the FDA.
Yeah, that's a really interesting question and great question. So food supplements, the first part is the supplement actually regulated by the FDA? Because not all supplements are. And so what I'm hearing you say is you actually feel that this supplement improves the daily impact of the disease on the patient's life. And so you would like it to be approved for this population as a treatment, is the sense I'm getting from your question.
Yeah. We expect very moderate effect, but I doubt it will significantly stick out if you do a really randomized trial. It will be hard to see something statistically relevant. And it's very tough to measure with our kids. It's really hard to write outcome measures.
Right.
So how do we approach that?
Yeah, so that's a great question. And I'm going to actually take out the food supplement piece there and just talk about in general, like how do we approach drug development, or how would we approach study design in this? And so I think first and foremost, you need understanding what do you think a product is going to do. right? So actually understanding what do you think it's going to improve, what aspects of the disease. So if we were talking an actual drug, we would have to understand that mechanism of action to be like, what is the drug actually doing? And then is you brought up the measuring, and with our heterogeneous diseases, which let's be honest, most of our rare diseases have heterogeneity. It is something that we combat every day. We think about this every day. So there are many approaches that we can take, such as number one is understanding what do we think is improving. And that is really going to be tied down. And so let's not think about modest yet, or it's not going to have a big effect, but what do we think it's actually improving? That's going to kind of help us think about how we're going to measure, because that's what we want to think about. So we do have to understand, what do we think we're improving in the patient's lives, so we can figure out how to measure it. We understand heterogeneity, and we have some approaches how we can handle that, but we still have to have some sense of what do we think is improving. And that's where understanding what the drug is supposed to do will help us in that. And then we can then help figure out how to minimize the heterogeneity aspect. So I know it's not the exact answer you want. It is not a simple answer. We have to really consider a lot of things. And one of the big things is understanding what do we think the treatment's going to do, and understanding what exactly is the treatment impacting a physiological or biological way of the condition. And then in terms of something for an approval for a food supplement, I would have to actually think about how that falls, if that comes into what center. Does that go to food per se? Does that go to the CEDAR for drugs? I'm not actually sure of that piece, but in general, we want to think about the aspect of what we think the treatment's going to actually do, and then that's how we're going to help us. So it is, it's not necessarily a simple answer, but it is something that we do face every day. But we have to look at a couple things, and perhaps when Dr. Panzara talks, he may have some thoughts about this as well, on how a sponsor approaches some of that in the general sense of with this heterogeneity aspect.
Okay, thank you.
I think the L3 is already approved. We actually buy American L3 from the GRIN store, and the label says FDA approved pharmaceutical quality. So I think that's already approved.
Yeah, it's safe, but it's not funded, so it's not.
Funded is a whole different story.
Thank you so much, Dr. Campbell. If everybody could join me in thanking Dr. Campbell for her time. Next, I would like to welcome Dr. Michael Panzara, the Chief Medical Officer of GRIN Therapeutics, which is our platinum sponsor for this conference. So thank you, GRIN Therapeutics.
Thank you very much. Can everybody hear me? Yep. So thank you very much for inviting me and us, GRIN, to present. I'm going to just talk a little bit about, just first of all, introduce myself. Mike Panzara, I'm a neurologist. I've been, I left my academic practice now a little over 20 years ago to enter industry. And because the thing I enjoyed most about my academic practice was doing clinical studies and trying innovative ways of developing drugs. So for the past 20 years, I've worked at a few companies where I have had actually a privilege of being able to get several drugs to the market with my teams. Of course, that's not something that gets done alone, as you'll see from the industry perspective of how drugs get developed in a moment. But I've been really privileged to do that. And I've worked in multiple sclerosis primarily, Duchenne muscular dystrophy, Huntington's disease, ALS, frontotemporal dementia, tough diseases with extremely committed patient communities. And it's that aspect of drug development that I never really knew of in academics. It's not something that you realize how important it is, the number of stakeholders involved in developing a drug, but in particular, the patient community. And I've been very privileged over those years that each of my experiences, I've gone through like an adoption process where I've been adopted by the community as a card-carrying member for a period of time. And it's those relationships that have enabled me to be successful and have enabled us to get drugs developed. So I'm new to this community, so hopefully I'll be okay to be adopted by you eventually, once you get my trust. So anyway, let me talk a little bit about GRIN Therapeutics, and our company, I'll just tell you a little bit about who we are, because we haven't existed very long. And so I think when a company's new and doesn't exist, I'll just have one slide on this. Then, Dr. Campbell and I didn't coordinate our talks, but she gave a beautiful description of what happens at the FDA. I'm going to give you a view of it from the outside really quickly. There's a lot of overlap. And by the outside, I mean from the industry. But as you'll hear, the interactions between the regulatory authorities and industry is a partnership. And it's very important that it's a partnership, because we each bring a scientific perspective to this process that makes better drugs, better, safer drugs. Talk to you about what we're doing specifically and the status of our clinical trial in Europe. So GRIN Therapeutics we're based in New York City. Nervati Neurosciences, of which I'm also the chief medical officer, we basically oversee the company. We have one single investor. Our approach is to fund a drug, make it private, means it's not in the public markets, and then bring it fully through to approval, fully funded through approval, so that external forces don't interrupt the drug development process. Because sometimes in the public markets, a drug doesn't have an easy time getting funding and make it to market. So that's our philosophy. Radiprodil is being developed for GRIN-related disorders with gain of function. I'll talk a little bit about that. First clinical trial is ongoing in Europe, and we just recently dosed our first patient. The company history it was inspired from the 2019 GRIN conference at Emory University, and then that sort of led to an interest in this area and the science behind it. And then in 2021, a leadership team was formed. And, we are, as I've mentioned, my approach has always been to partner with the disease communities to do this. And we have been, from the beginning, working with both academic and advocacy to make sure that we can do the things that are needed to be done in the way that the community needs them done. So how does a compound become a medicine? You already saw a great description of that. This is exactly what Dr. Campbell showed, but in a flowchart form, but essentially shows the process. She took you through most of this. I'm going to start sort of just a little bit more in the industry, what the content of some of these things are. I'm going to start at the preclinical development phase, because that's essentially where we began. And you end up with this part about what is preclinical development. Two components: the pharmacology, which means what does the drug do in animals? How does it act in animals? Like, how does it move through animals? What are the levels that are active at whatever effect you're looking for? And then the toxicology. What are the levels that are too high? The levels that lead to potential risk. And we do this typically in two species of animals, whether it's rodents, dogs, monkeys, to determine the highest dose possible before adverse events occur. And then we try to figure out what is the target dose that leads to a biological effect in the animals that would be predicted to give a biological effect in humans. So a lot of work here, a lot of time spent, and this is after. I'm not going to cover all of the innovative work that leads to the discovery of the target and why it might work in the disease. That's already been done. Now we're getting into trying to figure out how to do this in humans. Then we go to first-in-human testing, and the first-in-human testing, that could be healthy volunteers, that could be in patients, to determine that the expected biological effect that we saw in animals, what we predict happens in humans, happens in humans. What we predicted from animals actually happens in humans. So what is the effect of the drug on the body in terms of safety? And then how does the drug move through the body? How is it, once it's either taken or infused or injected, what happens to it? How does it go? How is it eliminated? So again, we need to understand how it behaves to basically expand into larger patient numbers and ensure that it's going to be safe and have the desired effect. So you'll hear phase 1, phase 2, you'll hear 1A, 1B. These are all different phases of first in human. And how we do it really depends on the disease we're targeting and what the question we're trying to ask in that early study. Then you get into the phase 2, which is really the first time you're trying to see, does the drug have a potential effect in a disease area? And that's called proof of concept. And that's often you do repeat doses. You actually always have larger patient numbers, because you're trying to figure out, when you give it to, okay, a few more patients, is it safe? in those more patients, are you able to start seeing your predicted biological effect? Because this is where you're really going to make a decision. Are we going to go to the next phase and try to get this drug approved? Because from an industry perspective, this is where the big increase in risk comes in spending, because you have to know you're going to have a drug that's going to work in pivotal study, or at least have a reasonable likelihood of success, because it usually requires a very, very large investment for the next phase, which we'll come up to. So you may test different doses, and you may do creative designs where you blend things together. And that's what you'll see here. Or sometimes we'll combine phases 1 and 2. Sometimes we'll combine a 1B, which means a multi-dose, with a 2A, which means a little bit of efficacy. Sometimes we combine phase 2 and 3. And this is extremely common in the rare disease space, because there are a limited number of people available to test the drug. So you want to minimize the number you have to use throughout the investigative process. And in a recent experience in working with FDA, we sat down for a rare disease area, and we entered into this program at FDA called the Complex and Innovative Design Program. And that whole program was geared towards taking an area of rare disease that you need to use innovative trial design elements, like minimizing, there's placebo minimization approaches, Bayesian probability assessments, all these new and innovative tools that are used to try and minimize the required number of patients that are necessary to answer the question about efficacy and safety. So this is where it gets really, in a rare disease, where it's very important to work closely with the authorities on an innovative approach to moving through the timeline quicker, because this could take a very long time. And then finally, you get to the approval phase. If you're lucky, most drugs don't make it, but if you're fortunate enough to get the end, then you have evidence of efficacy and safety. There's the process, and I only have the US and EU here, but Canada, Australia, all around the world, there are different processes to get this done. A standard new drug application takes between 8 and 12 months, depending on whether it's viewed as a priority review, which means adds a really, there's a very specific regulatory definition of what requires a priority review. So I'm not going to even try here because FDA is listening, but there is a path that actually uses that you can, in certain conditions, access this, or at least request this process. And then the EU has a MAA process, which is similar, and they also have accelerated processes that could be used. So this is another way just to illustrate sort of what's called the common technical document. This is sort of how all regulatory submissions are built. It's a pyramid or a triangle. You can call it whatever it looks like, but it's basically years and years of work from that discovery phase. And I didn't even talk about manufacturing, because you got to make a drug. It has to be done in a very high quality way. All of this builds up to a regulatory submission. And thankfully, the authorities around the world now have made a common technical document. So we just got to do one major format, with what's called module 1 being the one that's targeted for certain regions. So this submission is built from the ground up. You see all the different elements, details on the bottom, a little bit more higher level description on the top. The last one of these that I did was back when people counted pages. That was 4 million pages went into submission. I was telling people the other day, the fun thing to do 20 years ago was take a picture of the team sitting on the 18-wheeler truck that was driving the application down to FDA, right? Then that went to flying down a bunch of discs. Now it's all electronic, but it's just but just to give you the scope, 4 million pages. If you were to take 4 million pages and stack them on top of each other, right on top, it's taller than the Empire State Building. So it's about 1,300 feet. The Empire State Building is 1,250. I just looked it up before I came up here. So that's a lot of stuff. And what amazed me, and I'm still amazed by this is years and years of work, that somebody at FDA actually reads this, because and reviews it and has to assess safety and efficacy. So in terms of partnership, this is an example of, as we build this, we're of course interacting with regulators around the world as we are building this, but it does require a lot of work by a lot of people and time to get this right. So now that's it about the process from the industry's perspective. Spend a few minutes on why we're testing radiprodil in GRIN-related disorders. So this is an NMDA receptor, and this is the focus of the target of radiprodil. And so this is an example of the NMDA receptor with an NR2B subunit. You see that on the right-hand side. And this is not working right in GRIN-related disorders. I mean, the way that we like to describe this is, you have this gate of which controls the flow into and out of the, looks like the barnyard, I guess, here, where you have this flow in and out, and you have your chicks and your chickens on one side, and then the normal gate controls how much goes through back and forth. When you have a loss of function, gate's locked. Things don't flow right. When you have a broken lock, flow is unregulated and controlled. This leads to the symptoms that you're all too familiar with. And so our goal when we were designing the studies and thinking of how to approach this was based on all the research that had been conducted before us, we found that radiprodil actually has the potential to well, this just talks a little bit about, it's overactive. This leads to the symptoms you're all very aware of. And the goal here is to really trying to develop a specific therapy, and that's where radiprodil came in. So we realized that radiprodil binds to the NR2B part of the NMDA receptor. It's what's called a negative allosteric modulator. You'll see that around. That just simply means it's meant to bind to the receptor to lower that flow, to lower that overactivity. And the hypothesis that's leading us to try this is that by binding to this part of the receptor, you can reduce the overactivity, where you can have fewer seizures and improve development. That's the hypothesis behind it. That's why we're testing it. We have the phase 1b study that is currently enrolling in Europe. It's a phase 1b, but it also has a little 2a-ish element in it, meaning we're going to look to see if it actually has effect on either behavior or seizure symptoms. So it's evaluating safety and tolerability. We're also doing dose escalating to find out what the right dose is for that next study. We're looking for the impact, seizures, EEGs, other behavioral symptoms, other measures that may potentially be meaningful to patients and their families. And we just are right now targeting just people who have the gain of function variant of GRIN, depends on the country, 2B, 2A, 1, and age between 6 months, 12 years. And for seizure cohort, have to have some seizures. And behavioral cohort, we have tools measuring a certain level of behavioral dysfunction. So our posting in clinicaltrials.gov is still pending, and we thought it would be up by now. It just takes a little bit of time to get through the process. So the goal here, again, radiprodil at first in human, trying to look for a potential effect. So with that, that's really all I have in terms of the update from us. As we update our study information, we're going to make sure we're updating clinicaltrials.gov because that's the appropriate way to get the information out there relative to our trials. We are doing what we can to make sure we remain connected and that we're able to really know what's important for you. I mean, this is a picture from a meeting before I joined the company, just to show how we're trying to be to listen as much as we can to understand what is necessary. And that's actually related to why we're asking folks to visit our table, because we're also wanting to conduct a survey of what symptoms and things are most meaningful to all of you. Because again, as Dr. Campbell alluded to, there's an importance of whatever we're researching and whatever we're doing, and whatever radiprodil might do. It has to be meaningful to you. It has to bring something that you need, not in addition to seizure reduction, which everybody agrees is important. There are other elements of this disease that are even more important, that are not as well characterized. So we're trying to understand what those things are. And as we design, hopefully if radiprodil is safe and tolerated in this first study, the next phase, we need to understand what are the things that are important so we can assess those things and understand if we're bringing any value to all of you. So with that, happy to take some questions. Thank you for your attention.
Thank you, Dr. Panzara. Okay, so we have time for just a couple of questions before we transition to Dr. Tranfaglia. We'll open it up to the floor first. Any questions?
Do you have any other molecules you're working on for loss of function?
So not at this time. The way we have built the GRIN Therapeutics company is around, at this point, radiprodil, but our goal as part of GRIN Therapeutics is to look for additional compounds that we might be able to potentially broaden that portfolio again, to look at loss of function as well as other approaches to gain of function.
So it looks like there was a question in a previous session about why the radiprodil trial is only up to age 12 currently.
So, you know, whenever you design these trials, there's always going to be certain groups of patients that are left out of that first phase. And that is because when we are designing these trials, unfortunately, in some ways, you want to expose the minimum number of people necessary to answer the question, but you also want to expose a group that's narrow enough that you can see your effect without the variability of a large population. So when you have a large number of, a larger range, for instance, of ages, you need a larger study to be able to see a certain effect. So it's only where we're starting, the goal would be to expand. But this just so we can answer the specific question in as narrow a population is as necessary with a minimum number of patients.
So this drug, radiprodil, can inhibit only the defective NR2B subunits, or it blocks all NR2B subunits?
So we've actually run, so the question is, you know, does it only target NR2B or can it potentially impact other subunits? The data we have suggests it does impact other subunits, but through that binding. So it does other gain of function, other GRIN gain of function. It does seem to have an effect in animals. So that's why we've expanded the inclusion criteria to be beyond GRIN 2B. Underway in different countries as we move through.
Hi, Mike. I'm Jillian from GRIN Europe.
Yeah.
And as patients advocacy organization, we of course come into contact with a lot of researchers, and we also come across interesting projects where we think this would be something investigating further. Would GRIN Therapeutics / Novati Sciences be open to discussing future projects with us?
Of course. I mean, you know, I think that our goal is to build GRIN Therapeutics into a company that is focused on addressing the unmet needs in the broader GRIN population. We have to, of course, assess all of those in terms of, you know, how much risk and all those things like we would anything else. But of course, we're always open to partnerships in that way. That's how I've always done that in the past. Some of the best opportunities often come through patient organizations.
Thanks a million.
All right, thank you so much, Dr. Panzara. Next, I'd like to welcome Dr. Michael Tranfaglia, who serves as the Medical Director and Chief Scientific Officer of FRAXA Research Foundation.
Thanks a lot. I think my mic is on. All right. I'm Dr. Mike Tranfaglia, and for the last 29 years, I have been the Medical Director and Chief Scientific Officer of FRAXA Research Foundation. Our tagline is finding a cure for Fragile X. I'd like to talk to you about community-based drug development. This is a rather recent concept and a rather new name for that concept, but really it's what we're all trying to do. I want to talk to you about how you can get treatment strategies ready to take to the FDA and how you can get treatment strategies ready to partner up with pharma companies. So how rare disease foundations can take a part in the drug development process. This is something we've been doing for a very long time. And I guess you could see that either as an inspirational story or a cautionary tale. I'll let you be the judge. I am an expert in this subject. If you define an expert as a man who's made all the mistakes which can be made in a narrow field, because I've made all the mistakes you can make in this field, and I'd like to tell you all about it. First, I'll tell you a little bit about FRAXA. We're a nonprofit organization that was founded in 1994 by me and my wife. So that's how I got to be the medical director. I got in on the ground floor. We're run entirely by Fragile X parents, primarily me and my wife, but we've got a few other folks that work part-time, and they're also Fragile X parents. And we've got a board of directors that are, guess what, also Fragile X parents. So we're dedicated to funding biomedical research. This is why we do community-based drug development. We're not doing advocacy. We're not doing awareness. We do those things along the way, and we think we do more of that because of our efforts to actually develop treatments for Fragile X. We funded, actually, by the time we get through with our current grant cycle, we're going to probably be right about $36 million in direct research grants. So that's not just our total revenues, but the amount that we've actually given away in biomedical research. So we're founded on the principle that Fragile X is a treatable condition. And I know you folks feel the same way about GRINs. And I think this is fairly obvious now that we can do something about rare diseases. As we've heard from the FDA folks, they're approving drugs for rare diseases based on this kind of model. But this was pretty controversial when we first started out. And there were a lot of people within the community, Fragile X parents and family, who kind of got angry at us for suggesting the possibility that this was something that was a tractable problem. I think it's the experience we've had over the last few decades has borne out this proposition. And I think most people feel like this is in fact a tractable condition. So check out the website if you get a chance. You can see what we're up to. I think it always helps to compare notes and see what the other guys are up to. That's why I'm here. We're doing this for the two most important reasons in the world. And their names are Andy and Laura. Andy's our son, who is now 33 years old. So we started this when Andy was a little toddler. We actually launched this project before there actually was a 501c3. We got started right after Andy's 3rd birthday. So we've really been at this now for a good 30 years. Andy's sister Laura also has a full mutation of the Fragile X gene, but she's a perfectly normal woman, 31-year-old woman. She's married, she graduated from college, has a house in the suburbs and a nice job. Perfectly normal in every way. Is she completely unaffected? Well, hard to say what she would have been like otherwise, but she's great just the way she is. So just a little bit about Fragile X, because I'm not really talking about Fragile X, I'm talking about this process. Fragile X is the most common inherited cause of mental retardation or intellectual disability, if you prefer the more politically correct terminology. Affects approximately 1 in 4,000 boys. 1 in 4,000 girls also have the full mutation, but about half the folks are like our daughter, and they really don't have anything wrong with them because they have another X chromosome. It's the most common genetic cause of autism. At one point, before the definition of autism was expanded, it was thought that up to 5% of the people with autism had Fragile X. As we've had more genetic counseling, the number of people with Fragile X has actually gone down, while the number of people diagnosed with autism has gone up. Fragile X has a pretty subtle physical phenotype. You can recognize it if you're a parent or if you're a doctor who's treated lots of kids with Fragile X, but most people miss it. They look pretty normal. Our son's a very handsome guy. Our daughter's a beautiful young lady, but they have a striking psychiatric phenotype. I'm a psychiatrist, I would say something like that. About 20 to 25 to 30% of full mutation males meet the criteria for a diagnosis of autistic disorder. That is what I like to call big A autism. And just about everybody else has an autism spectrum disorder, which I often call little a autism. So really, Fragile X is an important model for the study of autism. About 20 to 25% of males have seizures. Most of those folks don't really have lifelong seizure disorders. They may have a seizure here, a seizure there. Most of the time, the seizure disorders are pretty well controlled. Our son has the seizure disorder, and it's reasonably well controlled, but he still has breakthrough seizures, even on 3 different anticonvulsants. Most importantly, Fragile X is a true developmental disorder. It's not neurodegenerative. These folks have a normal life expectancy, and they don't really have a whole lot wrong with the brain, except that it doesn't seem to develop over time. So the basics of the actual mutation, X-linked, highly variable penetrance. It's caused by a trinucleotide repeat that it gets expanded over the course of generations. That leads to the gene getting shut down. The actual fragile site, which, if I can point to it here, helped in locating the gene, in identifying the gene during the Human Genome Project, because it's right where the fragile site in the gene is when you incubate chromosomes in a folate deficient medium. That's known as the Fragile XA site, or FRAXA. That's where we get our name from. Actually, we didn't want to name it Fragile X anything because we thought it's kind of a misnomer, and we don't want to name our foundation after a misnomer because we'll probably change the name. But as it turns out, it stuck. So we're still FRAXA, and this is still Fragile X. There we go. So that's the fragile site right there. So basically, it's the absence of a single protein that causes Fragile X, and the severity of the disease corresponds to the deficiency in FMRP. And we've had a knockout mouse model basically since we started FRAXA, since we came on the scene. And as you can see, about 1/3 of the people in this study of childhood autism rating scale at the University of North Carolina meet the criteria for autistic disorder. But then you've got this big fat part of the bell curve. These folks are all squarely in the ASD range. And then these kids are probably just too young and they haven't really developed the symptoms yet, but they will. So what are we trying to do? I think one of our former board members put it best, and no coincidence, he was an MIT guy and an engineer. And he said, this is not basic science. This is an engineering project. We're engineering a solution for Fragile X. And I really like that. This is an engineering project. It's not basic science. It's applied science. And we, tongue-in-cheek, refer to this as the FRAXA method. We fund basic research, which generates data about mechanisms of disease. We fund translational research, which picks up on those mechanisms of disease and tries to find therapeutic targets, things that you can go after to fix the disease, or at least some of the mechanisms. And then we also fund preclinical validation studies where we test specific drugs that actually hit those targets that have been found through the translational research. And if we're successful, we have things that might make good drugs that we can take into clinical trials. Sometimes we actually fund the clinical trials if they're available drugs and we don't have a corporate sponsor. But a lot of times this preclinical validation, really importantly, leads to partnerships with pharma companies. And at any given time these days, we have about 30 different active pharma partners. So we have a lot of different companies that we're working with, different types of companies, big companies, small companies, all the way in between. Some are looking at small molecules, some are looking at the more biotech approaches, the biologics, the ASOs and gene therapy. That's what it takes. Of course, this isn't the FRAXA method. This is just rational drug discovery. This is what every pharma company in the world does it, but we do it as a nonprofit, and we do it on a shoestring budget. So we do what we can, but we can't do everything. And I think this applies to you guys as well. There are things you can do, and there are things you should do, but you can't do everything, so you have to prioritize. This is community-based drug discovery. So when nonprofits do it to try to help their kids, community-based drug discovery. So let's talk about some of these steps. What is it that we actually do? And what should you be doing? Well, one of the most important things is to develop mouse models. In your case, many mouse models, but in our case, even with a single genetic mutation. And in our case, we have just one kind of mutation for the most part. You guys have lots of different disorders affecting a number of different genes, a number of different receptors. So you're going to need lots of mouse models. It's really important, and it's something you can't really skip. A lot of folks nowadays, because I talk to a lot of rare disease foundations and consult with a lot of people about how to do this. And a lot of them think, well, we're just going to bypass that with iPSCs and organoids, and we're not going to need any animal models because this is cumbersome. And the characterization of the mouse models is time consuming. But you certainly need to do these things. And fortunately, creating the mouse models has gotten to be quite a bit quicker and easier and a lot cheaper than it used to be. There are other animal models that are useful too, and I would not rule out fish, fly, rat, and well, I would rule out primate models because they're really expensive. So that's something you might get to eventually, but Drosophila, fruit fly models in particular, are really, really cheap and easy to work with. So you can generate all kinds of mutations in fruit flies and spawn thousands of fruit flies, as anyone knows who's left the bananas out on the counter for too long. You can generate a lot of fruit flies in a very short time, and they're a great way of exploring genetics. So don't rule that out. There are many different possibilities, and you may find that certain animal models suit certain disorders, but when it comes time to do preclinical validation, it's always nice to see that you can get a particular strategy to work in multiple species. Even if you do and I'm sure you're going to do the iPSCs and the organoids, patient-derived cell models, you still need to characterize these, and you need to standardize the protocols. One of the big problems in science that leads to the so-called reproducibility crisis is that everybody does things a different way, and that's especially true with stem cells and with even cultured cells in labs. People handle them different ways, and so you get completely different results. So you still need to characterize these and standardize things. Providing reagents and the mouse models themselves and distributing all of these things can be really useful. There was a time when there was a real antibody crisis in the Fragile X research community. The antibodies didn't really distinguish the Fragile X protein from similar proteins, and it led to a lot of confusing results. And so we actually funded the development of monoclonal antibodies and distributed them. That's the sort of thing that you can do that's really helpful outside of the actual direct funding of projects, making resources available to the research community. When it comes time for translational elaboration, there are a number of techniques that have proven to be really valuable. Electrophysiology, often looking at cells or slices of brain, is a really powerful technique because you can combine it with pharmacology and biochemistry. Imagine if you've got a little piece of a brain in a dish. You can just put anything in there with it that you want. You can manipulate it any way you want. And so you can explore the pharmacology of the problem that you're dealing with. In your case, you guys have some fairly straightforward mutations in receptors and in ionotropic glutamate receptors that lend themselves nicely to this kind of pharmacology experiment. So you may find some surprising results when you start looking at this, and then you want to expand it out into live animals as you work your way along. Obviously, everybody wants to look at proteomics, transcriptomics, and all the other omics, metabolomics, and all the rest, microbiomics. But survey these things. It's really important not to make assumptions about what it is that you're dealing with, because as I'll tell you later, we've had some surprises along the way. Just when we thought we had everything figured out, it turned out that we didn't have it figured out at all. And it's also really difficult to sort out the primary pathology that results from the mutation versus the downstream secondary pathology versus compensatory responses that you see in the brain. If there's one thing that the brain is really good at, it's good at compensatory responses. Whenever there's a disruption in one area, it will respond to try to fix that. And the one thing you don't want to do is identify a compensatory response and identify that as pathology and develop some treatment that's going to wipe out your compensatory response and make everything worse. That's happened before. It's also important to study the disease state, not just the normal function. So there's lots of research that's been done on glutamate receptors, for example, but you need to study your disease models in depth because you can't assume that the disease is just the absence of that normal function. It usually spreads out beyond that. And you always want to incorporate rescue attempts, rescue paradigms in your studies. In other words, you want to incorporate in the basic research and the translational elaboration an effort to fix the problem at that stage, because if you don't, then it may very well turn out that it's just a compensatory response. If you can't rescue it with the obvious approach that stems from the basic research finding, well, yeah, that's probably not a real finding. Let's see. The next stage, of course, is preclinical validation. So now you've identified a treatment target, you've got drugs, maybe they're available drugs, maybe they're drugs in development, maybe they're just reagents that you order off the internet, but you've identified drugs that might actually fix this problem. You've got to do the testing in live whole animals with a whole brain whenever you're dealing with a brain disorder. As a psychopharmacologist, I can just tell you the essential issue is that you're going to be bathing the whole brain in that drug. And you need to know what that drug does to the whole brain, because there are lots of examples of circuits and brain formations where a particular kind of drug, say a serotonin drug, might do one thing in one place and it does the opposite thing in another place. And so you need to know how the balance works out. So standardized protocols, oops, I'm sorry. I meant to hit the laser pointer. Standardized protocols are essential because, once again, different labs will do different things in different places with different methods. And not only that, but a lot of academics, when they're working on a particular disease mechanism, they might test, for example, they might do 20 different behavioral tests on the animals, and only 4 of them respond to their treatment strategy. And then that's what they publish. And they don't even mention the other 16 that didn't respond or went the other way. So there is a certain cherry picking that occurs. If you have a standardized testing protocol, perhaps what we do is we have a drug validation initiative, which is a lab which maintains a colony of Fragile X mice in the same place. So it's the same mice in the same place, and they do the same behavioral tests on multiple different treatment strategies, usually small molecules, but also other things as well. And so we can compare those results. We can tell what's working and what's not working, because a lot of things in the Fragile X mouse seem to work. If you do 5 different behavioral tests and 3 of them respond, the academic says, wow, this works great. We know that's actually not a very good response. We need to take care of 5 out of 5 behavioral paradigms if we're going to actually take that thing into the clinic. So we also found out, for reasons I'll explain soon, chronic dosing is important. Tolerance is common with any kind of brain disease. Carryover effects are actually ideal. You want to see that you give a drug, it works, you stop the drug, and there's been some actual overall gain. Things don't just revert back to the way they were. That's more of a symptomatic and palliative kind of treatment. So like I said before, multiple levels, multiple species, that's really good too. So have those models if you have them, if you can get them. So let me give you an example of how this works in Fragile X. We're going to flash back to 2000. Group that we're funding at Brown University by the superb neuroscientist Mark Baer and an excellent postdoc named Kim Huber, who's since gone on to become a renowned professor in her own right. We're working on a FRAXA grant to study this form of synaptic plasticity called metabotropic glutamate receptor long-term depression. So this is a form of synaptic plasticity where group 1 metabotropic glutamate receptors get stimulated, and that results in a weakening of the synaptic contact. LTP, potentiation, would be a strengthening of that contact. mGluRs, mGluR 1 and 5, cause a weakening when you agonize those receptors. So this process, they've been working on this for a while, this process was known to be protein synthesis dependent. And their theory going into this was that this form of synaptic plasticity would be abolished. What they found was that in a knockout mouse, you actually had twice as much of it. So far from being abolished, it was exaggerated. And this had immediate treatment implications because if you could then block this receptor, DHPG is the agonist, if you could block this receptor, you could restore this to this. And in fact, they did that in vitro. This is in slice physiology, where you dissect out the hippocampus and cut it into thin slices and do electrophysiology. But it has immediate treatment implications, and so we wanted to test it in the whole mice. So we did that. So this is a FRAXA method. We go to the preclinical validation, and we use the reagent, not a human drug, MPEP, which is the prototype of the mGlu-5 antagonist, or it's actually a negative allosteric modulator, but mGlu-5 NAM in the Fragile X mice. And we're doing the audiogenic seizure paradigm because at the time that was the most robust phenotype that we had. And it seemed relevant too. But it takes a lot of mice to do audiogenic seizures because a lot of them go into status epilepticus and they die, even though that doesn't typically happen to humans. In the mice, that did happen. But we saw that if you treated them with MPEP in a dose response fashion, they improved significantly. And you pretty much eliminated all the audiogenic seizures. This is just with acute dosing, single dose of MPEP, and then you just do this an hour later. And we saw the same effect on open field activity. They were hyperactive. And there was very little effect on the wild types. That's not significant, but at high doses, you do see a little bit of slowing the wild type mice. These are their actual litter mates that don't have the mutation. And the Fragile X mice slow down when you give them some MPEP. So that's great. I don't want you to get the idea that we just did that little experiment and all the pharma companies said, all right, we're on board. No, 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 no. No, it was another 5 or 6 years and several $1 million worth of preclinical validation and de-risking studies to convince pharma companies that this was something that was worth pursuing. But basically every pharma company in the world was developing mGlu-5 agonists for a wide variety of indications, for anxiety disorders, mood disorders, various kinds of dependency, cocaine abuse, alcohol abuse, gastroesophageal reflux, all kinds of different things, Parkinson's disease. So we thought, wow, these are impressive drugs that have a wide range of activities. And a lot of those are relevant to Fragile X. We finally made the contacts and started doing the studies. First company out of the gate was a small company called Neuropharm that picked up an older drug that had never quite made it to market, but turned out to be an mGlu-5 antagonist. But they unfortunately went out of business because their lead compound failed. Same thing with Seaside Therapeutics. They licensed a Merck compound that was a little bit more advanced drug. Their lead compound went out of business. But then Roche and Novartis conducted large-scale international clinical trials, spent more than $50 million on this. And it all came up negative. Didn't work at all. We never really got all the results from Roche, but the Novartis folks were pretty open in publishing theirs. And in fact, they got a dose-dependent worsening in the subjects in the clinical trials. So it didn't work. So is this a problem with the method? I hope it's not, because we've validated a lot of other treatment targets for Fragile X using these same methods. Although I will say we've also gone to more chronic dosing because we knew that there was a potential issue with tolerance developing, but the pharma guys basically just dismissed that and said, nah, you don't get tolerance to an antagonist. No, it just doesn't happen. Well, that's exactly what we saw in the clinical trials. So this is an iterative process. Thinking about it in engineering terms, we've got a development cycle. We know we have a problem. We do the basic research. We identify the mechanisms of disease. We do the translational research to identify the treatment targets. We test the specific drugs and hopefully get them into clinical trials. If that works, well, we just sail off into the sunset because we're all done. More often than not, it doesn't, and you need to spin the cycle again. And you typically need to spin the cycle a whole bunch of times, especially for CNS disorders. And I just wanted to let you know that we finally did get a break because one of those other targets was phosphodiesterase 4, PDE4. In this case, we have a specific selective drug that's PDE4D inhibitor. And you can see that preclinical results showed that this is your phenotype in open field, hyperactive knockouts. And you give them the drug, and it rescues that. Social interactions, deficient in Fragile X, give them the drug, and all of a sudden they're equal to wild type littermates, and so on. And the really cool thing is, you probably think I made some kind of cut and paste error here, and I just put this on twice, but this is 2 weeks of dosing. This is 2 weeks on the drug, then 2 weeks off the drug, and they're still rescued after 2 weeks. We get a really pronounced carryover effect. We also saw an effect on the spine morphology, the actual structure of the neurons. And of course, the best thing of all is when it translates in a clinical trial. And here it did. We saw broad improvements on cognition with the NIH toolbox, the ABC scale, this is the visual analog scale. The KITAP, which is a computer-based performance assessment, didn't show any changes really, and that has happened in a number of studies. We're going to drop this one. But the ABC scale, we saw responses which were positive across the board. That's this direction, but not always statistically significant. More importantly, we saw the same carryover effect, that the folks that were on the drug for 12 weeks, then off the drug for 12 weeks, roughly the equivalent of 2 weeks for a mouse, retained a lot of these gains. They didn't go back to baseline. So that was pretty cool. So we've done a number of things right, we've done a number of things wrong. But one of the things we did was to focus on the neurodevelopmental disorder. You may know that there are other associated conditions in carriers. We basically ignore those because we figure we can only do so much, and these are different diseases. We develop actual therapeutics. So I would urge you to swing for the seats. Don't just settle for awareness, support, or training. This is something that you can do. You can make a difference even if you can't take it end to end. And we also funded a number of resources over the years, not just funding research projects, the mouse models, distribution. We've got conditional knockouts and a number of different, even though we have one gene, we have a number of different mouse models to study that. Reagents like antibodies and MPEP, the stuff that I was talking about, we funded the distribution of that. We literally had a big old mayonnaise jar full of MPEP synthesized and just started passing it out at meetings. And there was no better way to get people to test your compound than to hand them some for free. But, oh, wait a second. Just a couple more things. Meetings, I'm sorry, I'm pushing the wrong button. Meetings are a great idea. We started out small with these Banbury meetings that had 36 investigators in an intimate environment. We expanded to FRAXA investigators meetings that had about 150 people in big conferences. So imagine something this size, but just with the researchers that we were funding. And then we got tired of paying for that, and the Gordon Research Conferences agreed to actually do a series on Fragile X and related autism conditions, which is ongoing now. And I would also put in a vote for repurposing of available drugs, maybe a rational repurposing. So you have targets of opportunity that emerge over time as you're studying these things. Go after them and do the clinical trials. I think this is a great way to get good at doing clinical trials. Contract research is also really important because there are sometimes things that the investigators just won't do spontaneously, but you identify them as a critical need. Well, put out an RFA for those things and fund them yourself. It's mostly not as expensive as you think. It's certainly a lot less expensive than just rooting around in the dark. So the regrets that we have over the years is that we focused a little bit too narrowly, and it's the paradox of getting results. We figured out right away what FMRP did, or so we thought, but it turns out it does more than just regulate activity-dependent protein synthesis. And not only that, but it's not just postsynaptic, it's presynaptic too, and it's doing something completely different in axons than it's doing. You just assume it's doing one thing, and we figured out the one thing it was doing. Nope, not at all. And then it turns out it's not just working in neurons, it's working in the astrocytes and the microglia as well. And then it turns out it's not just the brain, it's having effects on metabolism as well. So don't limit yourself. You want to think about all of these possibilities. And another thing that's important to keep in mind is that drug repositioning, so drugs in development, drugs that are not available now, is harder than we expected, because often failure is a prerequisite. a lot of times companies won't allow you to work with their compound unless it's already failed for something else. And that was the case with the mGlu-R5 drugs. They'd been through trials for anxiety disorders and depression, and they'd failed in those trials. And then they said, okay, you guys can have a shot at it now. So now we're using a drug that we know doesn't work. And that's a problem. But then again, the tetra drug that gave us those nice effects that I showed you, the phosphodiesterase, that phosphodiesterase inhibitor was in large-scale trials for Alzheimer's disease. And they just took a risk on Fragile X because we had really compelling preclinical data and a really nice package to show them. So drug repurposing is really powerful, and you should consider it at every opportunity. You guys have the advantage of having some potentially very druggable targets. Glutamate is not an easy target, but at least it's there on the cell surface and we know what we're dealing with. So thanks very much for listening, and happy to entertain any questions, and hope this helps you to kind of think about how you want to plan your strategy in the years ahead.
So now we'll open up for one question on the floor, and we're running a little bit behind, so we'll take about a 10-minute break and be back in here for Dr. Keith Kaufman's next keynote.
Am I on? Okay, that was an excellent presentation. I think you probably started before the era of gene therapy, and a lot of research into that. Have you shifted your goals and strategies because of that development, and how has that worked?
It's a good question, because we actually started right during the first wave of gene therapy, and that was what we assumed we were going to be doing. So we started when Genzyme, which is located not too far from here, was doing protein replacement for Gaucher's disease, and they were working on gene therapy protocols for a number of different disorders. And we thought, we don't need to do this whole foundation route. That's a real pain in the neck. Why go to all that trouble? We could just talk Genzyme into doing all of this stuff for us. So we tried doing that, and we actually got a lot farther than we probably should have. They were very nice and they listened to us. But then, as you probably know, there were a few disasters clinically that happened in the early round of gene therapy. So in the early 90s, not only that, but there was this whole issue around healthcare reform, and there was a lot of uncertainty in the biotech investment market. And so we got the impression, and we got the message from Genzyme that, you guys need to characterize these models a little bit better, understand the mechanisms of disease, and just do a little work on this, and then circle back to us. So sure enough, after 25 years or so, we did circle back to these folks. And we are actually working with a number of companies that are interested in gene therapy. But also, because we've characterized the actual mutation well enough, we've identified targets for ASOs as well. So for those of you, you probably had some presentations already on antisense oligonucleotides. They're great for knocking things out. They're not so great for turning things back on. So we're missing something. So we want to turn something back on. Well, it turns out that there is they call them poison exons in ASO work. So there is a missplicing event in Fragile X that results in a mutant splice variant that basically gums up the works. And if you can make an ASO to that mutant splice variant, you can just turn the gene back on. And you find that in about 75% of the people with Fragile X. So we've now got a company that's launching that's going to be doing ASO therapeutics, but we've also got 6 or 7 companies that are interested in gene therapy, and a couple of other companies that are interested in other kinds of gene reactivation and protein replacement as well. So we like to call these definitive therapies. And when we had this ASO breakthrough, we decided we were actually going to hold a special meeting. So we're holding a special meeting at the end of April for discussion of these definitive therapeutics and how you're going to develop them. For example, we have a knockout mouse. The mouse does not have a trinucleotide repeat expansion. The mouse doesn't make this poison exon. It doesn't make any exons. It produces nothing. It produces no mRNA whatsoever. So we've got to study this in other systems, but we still need to make some kind of animal model as the final validation step for this. So that's one of the issues that we have to contend with now because of this breakthrough. So even after 30 years, these things pop up that you didn't anticipate, but it's good. It's serendipitous, and we're really happy about that.
Everyone, if you could please join me in thanking all of our speakers this morning. If you have additional questions for Dr. Tranfaglia or Dr. Panzara, please find them and ask questions during the break. We'll return at 11:05 for Dr. Keith Kaufman's talk.